protein translation. Initiator set is designed to apply
you can see it, I
weightings
can't.
and probabilities
You
to
can't see
any
the video?
mRNA sequence.
Pause, please.
No.
The
I'm saying
most
I
likely
can see it. Sorry.
proteins.
No, the video. The video.
Oh.
Yeah, I can. I mean, is it
can
playing for you
see
guys
it.
already?
Yeah.
Yeah, it was playing for us. Yeah, and we paused it for a moment because we thought you couldn't see it.
Okay. No, go on. I think in my case it might lag a bit.
You
But-
know, I think for
To
both of us,
calculate
but well, actually for all of
most
us.
likely proteins to be
What
produced
second are
from
we
this.
in? Initiator set,
It is
extendable open
also
source?
designed in the longer term to be possible to
yeah?
run in reverse.
Initiate a set demo from
And so,
Vulpine.
Okay.
to figure out
Video. Yeah.
which mRNAs lead to a specific protein sequence. But for now, its purpose is to figure out which proteins come from a specific mRNA. We will start by selecting a FASTA file, and this will come from wherever you've downloaded it to. And the FASTA file is
Have you posted a
format that is intended
040?
to just provide a plain text,
No. Wait.
very loosely
Did you see
defined
it post?
content for
136.
any.
Sorry, what?
Oh no, it's on again.
What are you talking about?
It's 136.
Sorry, are you talking about on the work adventure or on some other way of watching it? We're watching it
No, I
through
was
Work
talking.
Adventure.
Oh, you guys are watching it through. Okay.
Oh no, I'm watching it in VR, but like she's watching it in Work Adventure. But I mean, it should be the same.
No. I'm. But yeah. Okay. Just ignore me. I might be a bit lag, but yeah, don't worry.
Okay.
Alright. At the moment,
Genetic code.
I'm
In this
opening the
case,
FASTA file.
we're using the gene bag1. This FASTA file contains several sequences, including UTR sequences and CDS sequences that we've already made separate files of here for the purposes of our tests. But you don't need to do that. Here we're just using the whole sequence, the whole file, because now the MapAIC submodule, it is able to pass the cDNA and provide an mRNA sequence from that by converting the T's to U's. We can now select the data that we're going to use to apply probabilities and weightings to the mRNA sequence. Do bear in mind that in the production version, the future versions of Initiator Set, there will already be a gradient of probabilities applied as soon as you load your gene to the mRNA sequence that is to adjust for the actual position of the codons in the mRNA in the first place. But after that is where our selection of sample data comes in. We're using Lee et al. 2012 to adjust our AIC likelihoods based on which codons are actually there. And when we click Analyze, it will actually provide the color coding pattern for our mRNA that indicates what the probabilities are, and that is also provided in the numbers below. In the production version we will also include a probability waveform display mode, which unfortunately has not yet been coded, but it will be soon, and that will give you a much neater view from the beginning to the end of the mRNA as to what the changes of the
So like so you have like the mRNA sequence there, but like so what are these probabilities representing? Like
You're talking about the numbers. The numbers in the box in the middle of the screen are basically the probabilities that are being applied to each of the codons, and these codons are overlapping each other in all of the 3 reading frames. So it's
okay.
just based on the starting letter of each of those codons, which is why you can see the color coding has been applied per letter there, because it's saying the codon that starts at this letter gets this likelihood because it's this codon, and you can actually in the production version change the colors that are associated with each of the codons. And so those likelihoods then are listed below just for the sake of, you know, accessibility of the numbers if we need to work out exactly what's happened there. And like I was saying in the video, we'll turn that into a waveform, so as you get a nice graphical display that makes it more easy to see where it's more and less probable at this point that a particular codon would be used by a ribosome to start producing a protein. So that's kind of what that screen is about so far. It's the alternative initiation codons. And
Wait,
how likely each
so
one are,
when
because
when a ribosome is reading like an mRNA, like is it not just like reading them in like chunks of 3, one after the other, or like is it doing some, or like
it is reading it in chunks of 3, one after the other. That is indeed what codons are. However, it doesn't necessarily start from the very first codon, or even the first codon that looks very likely to be a start codon. There are many different factors that adjust the likelihood of a ribosome starting from a particular place in the mRNA and producing the protein from there onwards, up until it finds a stop codon. So that is what this software is meant to decode. It's meant to figure out what these different adjustments of the variables do to the likelihood that the ribosome will start from here or there in the code.
So really, the variability is like where it starts and where it ends, right? So it's like,
yeah,
it's like you should have like a probability of like, probability of it starting at different places, and then like, given where it starts, the probability of it ending at different places.
Well, the probability of where the end codons are is typically a matter of which is the next stop codon that the ribosome will encounter if it continues from this start codon.
Oh, so that one is more deterministic then.
Yeah, but the more probabilistic thing is where does it start from in the first place. And that can technically it can be just about any of the codons, except for the stop codons themselves.
Are these probabilities then the probability of it starting in different places?
If you look at the data list in the bottom of the with the coloured list at the bottom, that list there is scrollable on the actual software, and you can see what the probability settings are. And the ones that I put in there that I just selected a moment ago in the explanation of the video are from Lee et al. From 2012, who did a bunch of ribosome studies to work out what the likelihoods are of the ribosomes usually starting in particular places.
Okay.
Of course, there are many different
and this thing
where
this
that
thing
might
just
change these numbers slightly.
Like this thing is just like, sorry, like the immediate thing is just like that table applied to this, right?
Yeah, the color coding is those probabilities from the table at the bottom, being applied as these numbers in the middle to create the color code at the top, and the numbers in the middle are really what it's processing. So those numbers then are what it's using as the map of probability of that mRNA's different codons being used.
Yeah. Okay.
So then we can go through that again with the other tools that the video goes on with the explanation of them.
Cool. I think I get it. Should we? If no one has more questions, we can do.
Probabilities actually are. So, besides that, you'll also be able to change the colours that are displayed for particular codons. So you may set some of the codons to not show a colour at all, or you may just set some to show a very different colour to be really obvious compared to the rest. So, next we will run this through the Kozak calculator, and the Kozak calculator allows us to select a set of Kozak consensus sequences, probability weightings for them, and you may want to adjust those based on the species, based on the cell type, etc. Likewise, the conservation threshold, etc. as well. So these are all adjustable numbers. You can type in whatever you need. and then save those to a different file. We are going to now click Analyze on this and see that we have a set of potential initiation codons that are the most likely. We have here a set of Kozak consensus sequences now, highlighted by the importance of the letters according to the weights that are provided.
Huh.
And this enables us to see where we're most likely to get our protein translation from in the mRNA. So then we can go to the leaky scanning submodule, calculate whether there will be leaky scanning. In this case, apparently there will. And that's when the ribosome is going to skip certain parts of the mRNA in favor of others. We can translate to a protein sequence, and we can see that there are 9 possible proteins actually being shown here, not all of which make a whole lot of sense. Some of these Kozak sequences.
Just want to say, like, what is a Kozak sequence?
So Kozak consensus is the if you see those red highlights there, the letters in the mRNA code before and after the initiation codon that you're interested in, those letters will those codons will make it easier or harder for the ribosome to grip and actually use that as a starting point. So it's a very, very specific set of the codons of the nucleotides in the mRNA before and after the codon in question. And it was Marilyn Kozak who worked out that this sequence existed, hence the name. However, what she got wrong was that she thought that only some of the codons in the whole kind of alphabet, you know, the amino acid alphabet, she thought only some of them were actually possible to use as start codons. Turns out she was wrong. Most of them actually are, with the only exceptions really being the stop codons.
Wow, okay, I see. And so these surrounding sequences like basically affect the probability of the different start codons. Yeah. Huh.
Yeah, that's right. So that's all to do with how the ribosome actually gets a grip in order to, you know, begin properly using tRNAs to produce its protein.
Yeah, I feel like one general theme in biology is that most things are probabilistic at the end, right?
Yeah, that's what it turns into, a bunch of probability weighting. And that's what initiator set does. And you know,
cool. Oh, sorry.
Sequences overlap one another, so they may affect.
As I was
the
trying to say, you
likelihood
notice
as well.
in leaky scanning here we've got a bunch of different proteins, and not all of them make sense. Some of them are very short, but that's because you can see some of those initiation codons are very near the end of the mRNA. So even if the ribosomes produce a whole bunch of these short proteins, they might not be able to actually do much because they're so short.
And so they may
So,
just
you know.
end up being degraded or something.
Yeah. They may be used, for example, in the major histocompatibility complex to give the cell something to present to say what it is. You know, that's
I
one
see.
example of where it might still be used. But yeah.
Cool.
Usually, though, it's if there isn't a particular sequence to it that folds well, then it's probably going to degrade fast. Yeah.
Okay.
We could then move on to the remaining submodules. Please bear in mind that so far at this demo stage, Map AICs, Kozak Calculator and Leaky Scanning are the working submodules. Everything after this point is just a placeholder preview to give you a sense of what will be in the full version. So the mRNA localization is a submodule that will enable you to figure out where the mRNA is localized to, whether it be into the endoplasmic reticulum that the ribosome is then producing a protein, or into the cytoplasm. And the factor of whether the ribosome is studying the ER or not that may affect the way that it actually passes the mRNA.
Oops. Wait. Video.
Yeah.
Oh. Oh no, it's back. Sorry, like
What's
just
up?
VR chat just had a glitch, so you couldn't see the video. But it's back.
Well, because of its proximity to that ER membrane. The GC count will enable you to figure out how many Gs and Cs are present in the mRNA and what the importance of those actually is to the way that the mRNA folds and how floppy it is and how sticky it is. Submodule will be quite an interesting one, I feel, as it will enable you to adjust the bond weaknesses and compounding effects, the impacts that those have on the probabilities, and
So like the mRNA location, that's so it can be inside the ER or just in the cytoplasm.
well, that would be where the mRNA gets sent to the ribosomes. Are they in the ER in the ER membrane rather, or are they in the cytoplasm free floating ribosomes?
And
So you get
that's
both in any given cell.
and that matters
Sorry, I'm just trying to open the window here.
oh no, and that matters like
Well, it matters because any proteins that are then produced from the ribosomes at the ER membrane get pushed into the endoplasmic reticulum, which then gives you the pathway to proteins that are being expressed on the cell surface or ejected from the cell altogether.
all right.
Whereas the cytoplasm ribosomes are producing proteins that stay floating around inside the cell's cytoplasm.
Oh, okay.
And yeah,
Huh. And like what determines what mRNA gets into the ER membrane or into the cytoplasm?
the code of the mRNA, basically there's a sequence, a signal
Oh.
that says whether it should be picked up by the type of ribosome that is attached to the ER or the type that is floating around in the cytoplasm. And they're very similar ribosomes, you know, not much difference, but
Oh, wow, cool.
the signal sequence is still picked up.
And the GC count that determines how the RNA folds, or like there's some properties like, and those.
Yeah, that's coming up.
Okay.
By the way, Alpha, could you move a bit like here, like
We'll
closer?
enable you to
So
see
yeah, so
how
I
many
can like
GCs there are per coding sequence.
see the screen like better,
comparing
but also
between
hear
different
you
possible
closer.
proteins that you get from your mRNA.
Sorry,
Oh.
thank you.
Then
Go
we move
on.
on to the motif predictor. This allows you to check for things like hairpin loops, iReses, which are internal ribosomal entry sites, often used by viruses, and other motifs. The data sets that you can select from may come from external sources, or we may be able to include some based on prior calculations in the guess. Motifs that are found in this mRNA can be selected and viewed as ASCII art. That's an industry standard often used in a lot of scientific papers and textbooks. The UORF proximity, or
What's this noise?
untranslated open reading frame,
Oh, sorry. Me adjusting to the
handles
headset. Sorry.
the way that
Okay.
an overlap between one reading frame and another's potential proteins and initiation codons may affect each other. So you'll be able to sort the AICs by open reading frames, you'll be able to sort them by mRNA order, highlight untranslated open reading frames by proximity to major AICs, highlight UORFs by proximity to stop codons as well, be able to adjust
So,
the
like,
disruptiveness and impact.
Oh, what are ORFs like?
UORFs are untranslated open reading frames. I thought I said it in the video, but maybe not very clearly. It's when you've got like a protein that is encoded in a different reading frame, as in instead of starting with if you look at the sequence on screen, for example, instead of starting with UGU as your first codon, maybe you start with GUG as your first codon, or even UGA.
because those are overlapping with each other,
Where you,
but
are
they
you
are still
reading?
valid as far as. No, I'm sorry, top left, top left.
Oh,
Um,
like this part. Oh,
yeah,
okay. Oh, so you could start with like.
right at the top.
Oh, okay. Right at the. I was
Yeah,
looking at the highlighted
yeah.
ones in red, but
No, I'm talking about just at the start of the sequence there. As the ribosome starts reading through the sequence, it selects which reading frame it's going to use, and so it is either going to go with a reading frame that goes UGU and then GAC and then UAG, etc., etc., etc. Or it's going to go with one that goes GUG and then ACU and then AGG and so on and so on. Or it's going to go with UGA and then CUA and then GGA and so on and so on and so on. And those are
right.
all from the same few codons at the start, just overlapping each other just a little. And so what you've got there is 3 reading frames. And if you have a protein coding sequence in a different reading frame to the one that your ribosome is using, and if it starts in just the right place, it can trigger an untranslated open reading frame situation where it's not supposed to be translated, but because it's very close to the one that is supposed to be translated, it can disrupt that one being translated, or it can have other effects depending on exactly the way it's arranged. It can end up sort of stealing the ribosome into producing that protein instead or something. That's kind of rare. It depends on the exact coding. It's just something that we've got to take into account as a variable.
So you're saying that like a ribosome may start in the wrong reading frame.
Or in the right reading frame, but then be taken into the wrong one if it slips.
Oh, if it's like slips.
Um,
Oh, I see.
Yeah, and it's more slippery if there's a lot of G's and C's, for example. Hence the GC count.
And then it will basically produce a different protein. I see.
Potentially, yes. And so if there is a valid protein coding sequence that has a good start codon right in among your other protein sequence that is on a different reading frame, that can sometimes make things awkward and cause the ribosome to sort of stutter into the other code or just stop. Because it's sort of disrupted from the main sequence it was following.
I see.
So it can make a mess of the protein that you were making, for example.
Interesting.
It depends. Sometimes these things are deliberately inserted by viruses. So are internal ribosomal entry sites, or IRESs, which are a fold of the mRNA, or a motif, you could say, that basically forces the ribosome to start from a different place to where it would have otherwise started. And so that can make it so that if the virus has inserted a bunch of DNA, it forces the ribosome to always produce that protein sequence, maybe appended to the start of what would have otherwise been an ordinary protein, or maybe just instead of it.
I see. Okay.
So there's a lot of different variables going on here.
Yeah, I can see.
And apply them to the mRNA annotations, as well as viewing how they look in ASCII art. And of course the probabilities will all keep showing up in our numbers and our waveform display mode as well. Onwards to the ribosomal assembly, we can see the different ingredients of the ribosomes, from the EIFs through the rRNAs and tRNAs that they'll be using to produce proteins, and snow RNAs that may affect the assembly of them as well. We'll be able to calculate the effect of this combo on the likelihood of translation at each AIC, and generate models of ribosomes for viewing and export. The annotation matcher then enables you to bring together all of those previous submodules and their outputs into the annotation of the mRNA sequence. Be able to do a BLAST-P alignment to compare that with other proteins, whether those are local or from external databases. And you'll be able to also align motifs specifically to compare those motifs between different proteins
whether that's within the same mRNA or whether that's proteins from other genes or from other databases. And finally, you'll be able to do a functional comparison of the actual impact that these differences between the proteins that you can get from your mRNA and the differences in those differences that are caused by any mutations, etc. Those differences would be possible to compare here.
Oh.
And you'll be able to see the alpha helices, beta sheets, and other common transmembrane domains highlighted in the sequence view, and you'll be able to see a final combination of the probabilities displayed in the waveform and generate models of selected proteins for viewing and export. So that's initiator set. If you have ideas for further things to include, please let us know. If you would like to work with us to improve it, we'd certainly like to hear from you. And if you would like us to provide you with training for your staff, if you would like support, if you would like us to co-develop further systems and extensions to this with you, we're very interested to work with you and to see what you have to say. Thank you very much for your time.
And there's the music.
No, I see. Nice. Well, that's very cool. So this initiator set basically allows you to like calculate what's gonna happen with some RNA, like well, what proteins are gonna be made, basically.
And that is the kind of approach that we're taking to every aspect of how the body works. We're going with these kinds of probabilities and basically weaving them together and saying, okay, what does this actually do overall to the structure of everything? And how do all these variables affect each other?
But I mean, like I think RNA like translation is one of the processes that sort of is like sufficiently simple that we may be able to have like a very good model for. But if you then consider like all the other things that happen, like I mean, you know, like gene regulatory
You've not seen
networks
you've not seen our
and like
you've not seen our systems designs yet, have you? We know that initiator set is relatively simple. We do have much more complex designs for the other software systems that will handle these pathways and hormones and everything else, and all the the lipids and so forth. There's plenty coverage. I've had 14 years to think it out. Okay. I'm still thinking it out further.
all right, okay.
We've got a lot of things covered there.
Oh wait, Ribbon is having trouble. Oh no, I think this is, wait, just one sec. She doesn't know we're here, I think. No, I'm just saying Ribbon joined, but I don't think she knows we're here. Hey, we're here. Hey.
Oh god.
We're just downstairs.
Sorry, I forgot that we had jumped.
No, it's fine.
You alright, Kimber?
How about... I see. I mean, I saw this like cell model published this year that I think
It's not about
was trying to do like model basically both gene expression and, well, basically, yeah, like gene expression and like protein interactions, and also like, well, yeah, like how proteins regulate genes, like, and it was like trying to include like spatial arrangements and diffusion of proteins.
yeah.
Like
And
basically,
of course-
it was quite a, it seemed quite a complicated model.
Of course, any such model is going to be very complicated if you're trying to graphically, ray trace or whatever every single one of them. The intention that we have is to keep it as mathematical as possible,
So this is just about crunching the numbers themselves of how many of these proteins are likely to be doing this in that cell type, and so forth.
Well, but I think they were doing
So-
the special arrangement not for not for rendering, but like for the fact that it could potentially cause functional differences, right? Like,
Yeah.
I don't know an example, but like I think that was the idea. Like, but I mean, in principle, you could imagine it could have an effect.
I mean, another way you could do it is using voxels.
I
have each voxel,
I think that's what they did actually.
Represent. Yeah. You could have each voxel represent the position of a particular protein or molecule, or cell, or whatever level of zoom you want to be at in the body. And you could say, this voxel interacts with the surrounding voxels, and these are the parameters being adjusted as a result of that.
I think that's what they did, and actually they did have some kind of multi-scale thing where I actually don't remember the details because I looked at it several months ago. But
well, if you can find the link, then I'd like to see it for sure.
It was
just so I can compare it with what we have in mind.
Yeah, I think it was. Yeah, well, I definitely looked for it. Maybe remind me if I forget. But like, yeah. But yeah, they had like 3 scales or something. I think. Well, unfortunately, I can't remember what they were. But like, it looked quite interesting. And like at the lowest scale, they did have like spatial resolution with like voxels and like proteins being able to be at different states and like diffusion. And I think they were using Monte Carlo. Anyway, I don't remember all the
I would
things.
also be interested in checking that out. And like generally, this topic is really interesting. I hope we have more meetups on this.
Sure.
Thank you, Atha, so much for all your explanations. Like, it was quite advanced, like, but still, thank you very much.
Sorry.
So, like, it's
There you
I'm gonna
Oh, oops.
basically go to bed.
You take care.
I'm my grandma. Yeah, but you guys have fun.
That's good. Yeah.
And see you next time.
Thanks.
And I hope we have more.
Yes. Thanks for coming. Well. Bye.
No, I was trying to
Yay.
pet your ears, but oops, I just hit myself.
It's
But anyway, yeah, thank you. Hope to see you again.
Thank you.
See you. Sleep
wait.
well.
Thank you, Kemo, as well.
I am practicing. Wait. Okay, whatever. I need more practice for my. Yeah, like this.
Bye.
Take care.
Also, this was my first
And,
actual VR meetup.